I'm Loren Rieseberg. I'm a professor in the Department of Botany at the University of British Columbia. The goal of this project is really to develop vegetable varieties that are adapted to Canadian environments and to organic growing conditions. And that is really producing vegetable varieties that are more environmentally resilient. The way we do this is sort of twofold. First of all, we have what's called a participatory breeding program. And here we work directly with farmers to select varieties or to select individuals in our breeding populations that perform better under organic conditions. And then the second part of it is we bring in two sort of research components where we have a genomic component, which allows us to identify genetic variants that we can use to improve varieties. And then we also have an ecophysiological component that helps us to identify ecophysiological traits that will allow increased performance under organic conditions. And so together we use these to identify varieties that do well under organic conditions and also to improve them through breeding.
The Canadian Organic Vegetable Improvement Project, which we call CANOVI, is coordinated by the University of British Columbia, but also the Vital Family Initiative on Canadian Seed Security, which is a program of seed change. We work in collaboration with farmers, researchers, and industry organizations across the country so that we can trial regionally grown vegetables in farms across Canada in their fields directly so that we can see how these vegetables do in different conditions across Canada.
So as I mentioned, carrots are pollinated by flies and they have tiny little flowers. And so what we do is we put the carrots we want to cross into cages, and then we buy flies and we release them into these cages.
They're flowering a little faster this year.
Yeah, that's great. So these are the carrot cages. And this is where we have all the carrots we want to cross. So different cages have different populations. The selection is done on the farms, then they send us the carrot roots that they think are best, and then we combine them together to put them in these cages and cross them.
Have you talked, we're using this term population a lot, and maybe just to elaborate on that, you can see we have these different cages. And so within these cages is a unique population. And what makes a population is it has unique characteristics. So that cage has a selection of multicolored carrots. This one has a selection of red carrots. This one is a selection of orange carrots. And we don't want the orange and the red to cross-pollinate, so we isolate them. And we're isolating them because we're trying to do multiple populations in a single space. So that's why we've got these cages. When we introduce the flies, they'll only pollinate these and not pollinate these other ones.
The underlying rationale for this is that natural populations of plants and animals are well adapted to the environments they live in. And we can say the same thing for the land races of crops. As land races are early domesticated varieties that were grown by early farmers, whether they're land races or natural populations, have occurred in these particular sites for thousands of generations. And natural selection has optimized them for those particular locations and those particular kind of soil or temperatures that they occur in. So they're domesticated, but they don't have the modern agronomic traits that are seen in our carrots today. They've been grown for hundreds or thousands of years in a particular environment, so they're very closely adapted to that environment. We call them locally adapted.
In Professor Rieseberg lab, we are working in this project on organic carrot. We take this tissue, we extract DNA from them. And once this DNA is out, we check the quality and we produce libraries that are simply a collection of the DNA of each individual plant we have in the greenhouse. We expect to find genetic differences at the sequence level among those different varieties that might sometimes explain why they taste different, they look different.
And so the approach we take is we sequence these natural populations. We also sequence the land races. And we get a full copy of their genetic blueprint. We would be obtaining information for hundreds of millions of individual base pairs. And then we look for correlations between genetic variants or changes in those base pairs with features of the environment.
Here we are at the University of Toronto Scarborough, Department of Physical Environmental Sciences, entering the Plant Nutrition and Soil Fertility Lab. So our aspect of the research project really focuses on a concept called functional traits. So a lot of the research in crop biology has focused on agronomic traits. These are the things you see at the farmers markets. The color of a carrot, the size of a tomato. We're really interested in other characteristics of crops, such as leaf size or root traits. And these types of functional traits play a very large role in how well a crop will perform in various environments. Well, farmers are facing a lot of abiotic stressors right now. These are things such as high rainfall leading to flooding in fields, low rainfall leading to drought. Our research really contributes to understanding how crops can be resilient to these highly variable abiotic stressors. To do this, we focus on various traits that can confer benefits to the crop in the field and allow them to, if you will, strategize under various conditions to perform optimally under very heterogeneous environments. One of our main instruments we use is an infrared gas analyzer. This allows us to assess crop photosynthesis in the field. This instrument gives us data on such traits as photosynthesis, water use efficiency, and this really allows us to understand how crops are performing under different environmental conditions.
The best way to encourage the farmers to be involved in the project and to use these varieties is if we can generate varieties that outperform or perform as well as imported seed.
As part of the CANOVI project, we offer a variety of carrot tasting events across the country. And the purpose of this event is to bring consumers, producers, and vegetable growers together and get their opinions on how the different carrot varieties that we're working with taste like, cook like, feel like, look like. And the reason why we do this is because with any breeding program, you want to breed for agronomic traits so that it grows well generally, but at the end of the day, if no one likes the taste of the carrot that you're growing, then all that work can be completely for nothing. And we're hoping in a couple years, we'll be able to commercialize that variety as a completely open pollinated, organic, and non-proprietary variety that growers across the country can access and use for whatever reasons that they want.
I want to point out, first of all, that we could not do this project without Seed Change. Seed Change is the one who engages with the farmers. They also are the ones who find the different varieties that we use in the project. They are responsible for distributing them to the organic farmers. They also are the ones who set up the evaluation protocols and the selection protocols with the farmers. They also play an important role in communicating the results back to the farmers.
We hope that this work will expand to other crops, not just carrots, and with the work that Dr. Marnie Isaac is doing and Dr. Loren Rieseberg is doing, we hope that there can be a genetic basis and a functional basis for breeding different crops for climate resilience within Canada that can speed up the breeding process and make it more efficient so that we can improve and develop more crops to meet the diverse needs of farmers across Canada.